Hello, welcome to our webinar today. The Biochemical Society and Portland Press are pleased to welcome you to this webinar, part of the Biochemistry Focus webinar series. Topics in this series will include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please see the website for more details. I am Luciane Vieira de Mello, Professor in Bioscience Education from the University of Liverpool. I'm also the chair of the Biochemical Society Training Theme Panel. Today's webinar is called How to Deliver Biomolecular Science Honors Research Projects with Limited Lab Bench Time. As we all know, lab time and bench space will likely be limited for many final year undergrads due to social distancing, shielding, and travel restrictions. Therefore, projects will need to be adapted to this new landscape, whilst still achieving their aims and learning objectives. The webinar today is the first of a two-part series that aims to share the advice and expertise of supervisors and students with experience of such projects, and we hope that you will find the information shared useful as you perhaps look to adapt your own. Today, I'm pleased to welcome Dr. Douglas Houston and Vincent Blay Roger from the University of Edinburgh. Doug is a senior lecturer in computational biochemistry who supervises purely computational projects, including molecular dynamics, cheminformatics, a QSAR, virtual screening, and molecular dynamics. Vincent is a student in Doug's group who will be talking about his own project and undertaking it through remote learning. Questions you will be asked at the end of the webinar, but please feel free to send your questions during the talk. If you have any question, please type it in the question box as shown in the image on the screen. I now welcome Doug and Vincent for this webinar.
Okay, thanks for that introduction, Lou. So I'm going to tell you a little bit about how I go about supervising remote practical projects was mainly aimed at honours students, but it's also applicable to master's students, and I've also supervised summer undergrad students as well. So just a very quick bit of biography about myself. So Lou's done a really good job of telling you what I work on, but so I'm originally a protein crystallographer by training, and subsequently have moved more into the molecular modeling side of things. But all my work has always had a strong emphasis on drug discovery. So Lou mentioned that I have a lectureship in computational biochemistry. And what is computational biochemistry, you might well ask? Really that's look at using computational tools and approaches to try to either support or refute hypotheses that may be based on wet lab data. So I have a lot of collaborations with biochemists and cell biologists who run wet labs, and they often come to me and ask for various types of analyses that they can then include in whatever publication they're trying to generate. So that might be protein-protein interaction predictions, It might be predicting the structures of proteins through homology modeling or other means. Prediction of protein dynamics, usually through molecular dynamics, and I'll be telling you a bit about a molecular dynamics project that I supervised. Virtual drug discovery and screening is a very large part of my work, and as a subset of that, cheminformatics. So I'm quite interested in methods development, actually trying to improve the efficiency and accuracy of these computational tools that we rely on so much. So the particular honours research project that I'm going to tell you about in more detail was titled Computer-Aided Design Biomolecular Dynamics of Conformationally Stabilized Peptidomimetics. So this was a project that was looking at a particular protein target, an oncoprotein called SCF-SCIP2. It's an E3 ligase that ubiquitinates the tumor suppressor P27 for degradation by the proteasome. So P27, as I'm sure you're aware, is a very important cell cycle regulator, and downregulation of this cell cycle regulator has been identified in a very wide range of cancers. This particular E3 ligase, SCIP2, has been shown to be overexpressed in a large number of specific cancer types, and it's been shown to also upregulation of SCIP2 has been shown to correlate with reduced survival rates. So if we take a little bit of a look at the structure of this E3 ligase, so we've got the complete active complex on the left. You can see it's quite a large complex that's made up of a number of different subunits. On the right, we have the crystal structure of part of this E3 ligase complex, comprising the subunit SCIP2 and SCIP1, this F-box protein. We also have in cyan this CKS1 so-called accessory protein, and together the CKS1 and SCIP2 form a binding interface for the substrate P27. So the idea is that we would like to inhibit the function of this E3 ligase, and in so doing, hopefully have some sort of positive effect on cancer cells, either slowing their growth or ideally killing them. So a closer look at the structure of SCIP2 and CKS1, if we zoom in a little bit. Here on the left, we've got SCIP2 shown as surface representation, and you can see this concavity in the center, and this is actually the binding site for this CKS1 accessory protein. So if I show the CKS1 protein, you can see that it docks very neatly into this shallow binding pocket. So if I show CKS1, instead of a surface representation, we actually take a look at the backbone in the secondary structure view. We can see that there is an alpha helix just here. Hopefully you can see my mouse pointer. Just here that forms a large part of the binding interaction between the CKS1 protein and SCIP2. And if you strip away the majority of the CKS1 protein, which I've done here on the right, you can see there is actually an 11 residue long peptide, which makes the majority of these interactions. So then one strategy to try to disrupt this protein-protein binding interaction would be to produce this peptide and hope that it acted as a competitive inhibitor to disrupt this interaction. But a fly in the ointment is this alpha helical conformation. So as I'm sure you're aware, peptides don't tend to like to form alpha helices spontaneously in solution. but in recent years there has been a number of chemistries developed designed to stabilize peptides in an alpha helical conformation. And some of these chemistries have been given the nickname stapling. So we actually had a project back in 2017 with some synthetic protein peptide chemists who looked at using this olefin stapling strategy to lock this 11-mer peptide in the alpha helical conformation in the hope that it would be able to bind to skip2 in the CKS1 binding pocket. We did a number of analyses on these peptides that were synthesized via both circular dichroism and NMR, and that showed that indeed at least some of the staple designs that we came up with did indeed seem to increase the helical content quite dramatically. So here on the lower, and really just wanting you to look at this table on the lower right. The middle column shows that with no staple, this peptide shows no helicity in the CD spectrum, but with certain types, certain stereochemistries of staple, we do see an increase in percent helicity. The only drawback to this approach is that this synthetic chemistry is not particularly quick or easy to perform in the lab. It's not like trying to create derivatives or analogs of a small molecule hit, where you can generate a large library of related compounds and test them in an assay to see if you can increase the potency. That option isn't really available when you're dealing with these types of molecules. So the idea behind the honours project was to see if we could use computer-aided design to help our design of this stapling chemistry and to see if we could use the computer to predict whether or not certain types of staple would increase the helicity that we needed. So to embark on this project, what did we need to start with? First of all, we needed the data that I just showed you in the last slide. We're mainly working from the CD spectra, but that we also published the NMR structures for those staple peptides, which were a very useful reference. And of course, we needed the peptide sequences. In terms of tools, we needed a molecular dynamics package. In this particular case, we used NAMD, but there are a good half dozen other MD packages out there that would have been just as good. Things like Gromax, Amber, very popular. We used NAMD partly because it's extremely well supported and partly because you don't have to pay for a license. We also required molecular viewing software. So as related to NAMD, the authors of NAMD, they also supply a molecular viewing package called BMD, and we also make great use of PyMo as well. And then we also needed trajectory analysis tools to actually make sense of the heaps of data that a long molecular trajectory generates. So again, BMD has some built-in tools for this, and we used some other packages, WordDOM and CCP Traj. So that's it in terms of software. We also needed the hardware to run these programs on. So I had access to 2 GPU accelerated Linux workstations, which were quite powerful. And we were also allowed access to EDI, which is the University of Edinburgh's high performance computing supercomputer. And we also managed to get some time on Archer, which is the UK's national supercomputer. In terms of skills that the student needed to come with, the prerequisites were not too stringent. The student did obviously need to have a working knowledge of protein and really peptide structure and chemistry. But for this project, although it was purely computational, no programming skills were required, and I provided tutorials on how-tos on how to use Unix command line and all of these software packages. In terms of project progress and outcomes, usually these projects start with a lot of reading. I supply the students with at least a dozen or so papers, sometimes more, so they have all that to plow through. I'm now at the point I've had enough students working on projects like this that I can supply those students dissertations, which students really find very helpful and useful for, well, both for getting a grounding in what the project's all about and the methods they're going to use. It also gives them, you know, if you give them good dissertations, it gives them a good guide on how to, you know, what's really expected, what the final output is that's expected from them. As I said, I also provided a large number of tutorials and how-tos for the various software tools. So I basically, at the very beginning of the project, we throw all these things at the student and they kind of go quiet for a while as they work through all this stuff. I find that in terms of progress, it's usually highly dependent on the motivation and initiative of the student. It's not so dependent on the, I don't know, intelligence of the student or the skills that they might already have. Sometimes students come already knowing Unix command line. Sometimes they come already with a programming language, but I tend to find that doesn't matter so much. It's more about the student's own motivation and how well they can get on with things by themselves for a while. For this particular project, progress was initially slower than usual as the student had to do all this reading and got to grips with the software. The particular student wasn't so strong with peptide chemistry, so there was some brushing up to do there. But usually what happens is progress starts to really accelerate, hopefully by around the halfway point as things start to click. So for this particular project in terms of outcomes, the student managed to generate enough data to conclusively prove that the particular force field model that we were using, which was an implicit solvent model, wasn't actually accurate enough to successfully reproduce the wet lab data that we were using as a reference. So this meant that we could be sure that future work would need to switch to an explicit solvent model, which I was good to know because explicit solvent models require much more compute power. So for students starting on the follow-on project to this, I can now get my hands on rather more compute power, which means they'll be able to bring to bear a lot more resources to their project. So here, this table is just showing a few snapshots from the various molecular dynamics trajectories that the student generated. In yellow on the left, we have the unstable peptide. You can see that it spent basically all of its time during the simulation as an unstructured coil. On the right, we have some snapshots from the simulation of the stapled version of the peptide. You can see there is some alpha helicity. It is trying to form a helix, but unfortunately it wasn't quite enough to match the percentages I showed you earlier in the circular diacharism table. So in terms of skills acquired by the student on this project, they became really quite expert at the Linux command line, and in so doing managed to pick up a smattering of programming. So certainly personally the way I learned programming was during my crystallography days. I had to use the Linux command line, I saw that there were quite a lot of rather repetitive and laborious tasks that I had to do that really ought to be better done by a computer. So I taught myself some shell scripting, and if you get good enough with shell scripting, you really have learned a programming language technically, and that launched me into really learning real, so-called real programming languages properly. The student also got a working knowledge of molecular dynamics. So molecular dynamics, what it's good for, how to actually go about setting up a protein solvent system, because that is not trivial if you want to do it correctly, how to actually run the simulations, and then how to analyze the data and draw conclusions from it. And that part there is also not trivial. It takes quite a lot of thinking and manipulation of very large amounts of data to be able to draw convincing conclusions. As with, you would hope, most honours or master's projects, writing up any dissertation ought to teach a student to get much better at critical analysis and improve their writing skills a lot. And one of the skills which you might not first think of is that the student got really very good at knowing exactly how and when to ask for help. So what do I mean by that? So I've been supervising remote projects since around 2012 now. I think I've seen around 20 or so dissertations being produced, and I've found a big help when you're trying to supervise a remote project is managing the students' expectations and teaching them exactly how to ask for help. So that means that when they email you because they're stuck on something, which of course they often do get stuck on things, what you don't want from them is an email that just says, oh, it's not working, because inevitably then your reply to that has to be an email asking for more information, what's not working and in what way is it not working. So in our very first Zoom call, when the student is first starting the project, I show them this slide, how to ask for help, and just spelling out explicitly all the sorts of information they need to include in their initial query so that we don't end up in a back and forth conversation where I'm simply trying to tease out of them what's gone wrong and what is it they're actually trying to do. I think this is, when they get the hang of doing this correctly, it can really speed up them making progress and not being stuck on a particular obstacle. So in terms of what did and didn't work on this project, I mentioned these how-tos and tutorials already, and for molecular dynamics at least, especially for NAMD, there are a huge library of how-tos and tutorials already out there available for free online. And a quick Google can allow the student to find these themselves. So that, if you can if they can't find one, you yourself can find one and send them a link, and that really does save a lot of teaching time. They can work through that instead of you having to show them step by step how to do something. I've found that in general, and this might be slightly surprising, that email is actually more effective than remote face-to-face communication tools like Zoom or Teams or Skype or Collaborate. I do use all of these programs, but I find the asynchronicity of email kind of speeds up the back and forth of communication. So I never want my students sitting stuck on something, waiting for our regular Friday Zoom meeting to ask me, I would much rather they sent me an email straight away. And you know, if I'm not in my emails at that moment, then I will be soon, and then I can send them a solution, and they can then start making more progress again. So in this project, what worked? Well, our simulation did not match reality, but we did generate enough data to be sure why it didn't match reality. So we now have a clear path of what to try next time. So that was quite good. Something that perhaps didn't work in this instance is that, as I said, progress was a bit slow at the start, and perhaps I should have pushed harder near the start of the project. So some students are really good at managing their time and giving themselves interim goals. Some aren't, and when the deadline seems very far away, sometimes obviously the motivation can be rather lower than towards the end of the project. So in terms of general advice, apart from everything I've said so far, I think for a remote practical project, communicating with the student just absolutely as much as you can. As I said, mostly via email, but we have weekly Zoom meetings as well. That I think helps them feel a connection, stay connected to the project, and it helps keep up motivation and engagement. But you have to really try to manage to do that, but without being overbearing. So I know during lockdown, students were feeling anxious. They were struggling even more than usual with stress. So you definitely don't want to be contributing to that. And another thing I want to just bring to everybody's attention is this website, VLS3D. So this is an enormous library or list of computational approaches and free resources to assist, well, it says drug discovery, chemical biology, and precision medicine, but it really covers pretty much any biochemical question that you might be interested in. I have no affiliation with this website, by the way. I'm just, I have it here at the end of my presentation simply because I have found it so incredibly useful. So I urge you to go and check it out. There are literally dozens, if not a couple of hundred, of links to various web servers, software packages, computational tools that there will definitely be something there that could help you answer the biochemical question that you're proposing to turn into an honours project. So that's the end of my project. I believe Vincent is up next.
Fantastic. Thank you so much, Doug. So my name is Vincent. I'm a student in the group of Professor Doug Houston at the University of Edinburgh. And I'm just going to be sharing a bit about the project that I'm working on at the moment, most of it like remotely, and also some of the things that work and didn't work in this kind of remote distance research project that we are working on. And I have been collaborating with Doug for quite a bit now on a couple of remote projects. So it's a bit of a personal opinion. The project that I'm working on at the moment, as you can see on the right, there is a great component of computer-based work. But before that, as Doug was mentioning, we do quite a broad and extensive reading of the literature to set up interesting hypotheses and see where the frontier of current knowledge is. And hopefully in this project, maybe in the end, we will have a smaller component of wet lab experimentation to kind of validate those hypotheses or the best results that we were able to generate using a computer. And more specifically, what we are doing is in this project, first we are trying to identify targets, that is biomolecules in the human body, which if we were able to track and modulate their activity, then that potentially could have a beneficial effect on the health of people with certain disease. So once we identify those biomolecules that we want to design drugs for, then we can use computational tools to kind of start generating compounds that could be able to modulate such activity. And both of these components, the target identification and the design of new compounds, all of these we can do pretty much with the aid of computational tools. And then in the end, we will try to go on the wet lab and test whether these predictions, whether our best current hypotheses hold true when we do the experiments. And if everything works well, then hopefully we may identify some compounds that work at this very preliminary laboratory stage, and then that could be taken forward and do deeper studies, and hopefully down the line help someone and improve the health of somebody. And I think that is, in my case, something that's very important to me, that keeps me motivated long term, as Doug was mentioning. That's something I find important to keep yourself motivated. And it's something that I will come back to as I talk more. So now what I share is a bunch of actually 20 points of things that I found were very positive when my mentor did them, and that really contributed to our research or contributed to my satisfaction and engagement with the project and the research that we were doing. I'm not going to go through them individually, but I will highlight a bit some of them. And some are specific to distance projects, but others are common to whether you would be working remotely or you would be actually on a wet lab. So for instance, Doug mentioned communication. I think that's totally essential to set up an effective way of communicating. How to communicate these kind of errors. We're working on the computer. That's something essential, and that helped it so much. But email is still not as good as being next to the person and exchanging ideas, having an active dialogue. So I think you have to be also like understanding. Like email is more prone to also misunderstandings at some points. There is all of these non-verbal communication that doesn't go through. So maybe you need to be a bit more patient than usual, as having a person next to you and discussing things, and a bit more understanding that misunderstandings can indeed occur, and yes, more tolerant. Aside from communication, I would say like you still want to honor whatever values you are trying to teach your mentees about. And for instance, you want to be like really exceptional and in ethics or in any other values that you're trying to communicate to and teach to the people in your group. You still want those to go through even if you're working remotely, and I think that's something that's a remarkable job. The second list is more a bit about management. So indeed, when working in these distance projects, I think you don't get to see the rest of the team working as you would be in a wet lab under normal circumstances. So you have to be, I think, more trustful maybe of what people are doing, what they have to. And I guess if you are the mentor, you may want to be more vocal in terms of, if you have several people like responsibilities, who's accountable for what, who has to do what, and by what deadline, and so on and so forth. And like this trust, I think it's also a two-way street. So you want to, you want me as a mentee like to be comfortable sharing with Doug like what is working, what is not working, what I don't know, and just be comfortable to reach out. And I think one way to achieve that is maybe, at least for me, like feeling empowered. Like if one example would be if we are doing some research and I come up with something that was really unexpected, but I'm confident about the way we have done it, then I know Doug will trust me and say, okay, if we need to change research direction based on what you are finding, we will do so. So it's a way of really empowering your people, and if they come up with a better approach, we will use that approach, and so on and so forth. And lastly, these last points I think are a bit more about logistics. So even if you're working on a computer-based project, I think you still want to invest a lot of time in properly designing your experiments. What are the research questions that you really want to answer, and what is the best way of doing that? That's essentially equivalent as if you were doing wet lab experiments, and you still need all of that prior time investment in setting up, sound hypotheses and see what's really the frontier of knowledge. And then what's a big difference now is indeed the time that we have available to go on the lab and try different experiments. Wet lab is much more restricted. So I think what you really want to do is be selective and prioritize those experiments that really make your research advance forward. So yeah, just prioritize experiments, be very strategic. These are the kind of questions, and these are, try to anticipate what could be the possible results that you would get, and so on and so forth, to maximize that value of time in the wet lab. And one important thing also going about the trust and the communication, I think, is in the end you want to realize that both mentor and mentee and the rest of the team, we are all on the same team. So you want to, I think everybody should be invested in the project, pushing the frontier of knowledge forward as much as possible, and caring less about, where did the ideas come from, or what was the original design, who was it proposed by. It's a matter of, focusing on the results and making sure everybody stays on the same page and on the same team. So, any kind of ego, pride, or biases, you want to put all of those aside. And finally, some word maybe for the mentees as well, if there are some students, I guess, in the audience. So if at this moment you are, interested in doing some research, but you're wondering, like, well, now with the virus situation, maybe it's not a good time. From my experience in many labs, what you find is this data generation from a wet lab is often not the bottleneck. Many labs will have plenty of data as well available, and actually analyzing the data, modeling, building new theories, interpreting the results, that can often be way more time consuming than just doing the wet lab experimentation. So chances are you may have some possibility. So yes, reach out to your favorite professor and see if they have some research opportunities available. In the worst case, you will just be in the same situation you are, and you can try with another professor. But in the best case, you may have a wonderful research experience as I'm having. And the last thing I wanted to mention is that about motivation. So I think you want to work on something that's going to keep you motivated long term, like these computer-based projects. Don't think they are going to be like super fast either. They may take time, just a wet lab experimentation. So find something that keeps you passionate about. And in my case, as I mentioned at the beginning, was like knowing that something we are doing maybe down the road eventually could help someone and contribute to their health. And, everybody, you have to find your passion, I think, and find the opportunities, and I think they are out there. That's pretty much it. I think now we're going to reconvene and we will have some Q&A. Thank you so much.
Okay, hello. Thank you very much for the talk, Doug and Vincent. Fantastic. I think there is a lot for us to think about there. It was very nice to listen to the students' view. I value a lot the students' input in everything we do. So in terms of the questions, we will start with you, Doug, that spoke to us first. There are a fair amount of questions we have here. So what I'm trying to do here is to combine the questions. So for those who join us today, if I don't mention your name in particular, it's because instead of reading question by question, which would lead to a lot of repetition, I kind of grouped them in themes, okay? So Doug, one of the first themes would be the time for the project. We know different universities, sometimes a bit of the differences on the length of our honors project from one institution to another. So if you could talking a bit about the time your project, things happening in Edinburgh, but also, as I said, have in mind that other institutions may work a bit different. So what would be the minimum time?
Do you mean in terms of the length of the project? How long the? Okay, so I'm honours project students. They start in January, and then they finish up in around May, I think. The first half of that, they're still going to lectures and things, so they're sort of on half time for the project, and then halfway through that, they switch to going into the lab full time, or not, as the case may be.
And what is the expected time from the students? How often the students dedicate time to their honours project in the final year?
So, I mean, it's once they're on full time, it's sort of 8 hours a day, 5 days a week. Is that what was meant? So it's full time.
Yes, and presumably it doesn't make much difference if they work kind of, if a block, like some universities, they don't run lectures, the students only focus on projects. Other institutions,
Yes.
kind of a part-time, if you see what I mean. It doesn't really matter, does it?
No. I mean, you can. So as I said, the first half of the project, so January, February, they're only doing, I think, 1 or 2 days a week on the project, and the other days they're still doing other coursework. So I guess you could do that for the whole project and spread it out for longer. But yeah, it doesn't seem to make much difference in terms of this sort of speed of progress.
And I know I said the questions were for Doug first, but Vincent, as a student, would you agree with that, or would you have any comment?
Yeah, I mean, something interesting is that, the timing is also, or maybe different. If you're in the wet lab, you may be running experiments and they finish at, whatever time, and you may need to come to the lab. So with computers, it may be similar, I guess. Sometimes you launch some computations and you need to resume them, at whatever time that is. So, you may need to be pending of your, computational experiments in a sense, in a similar way. And in terms of the time dedication, I think working remotely is possibly pretty similar to being in the lab. You still want to do, your reading, you want to analyze the results. And as Doug was mentioning before, you generate large amounts of data and you want to really think how you're going to deal through all of that. So yeah, you can be working full time on them.
Okay, so the second theme would be support. And what I mean by support here is related to the students' prior knowledge to take a computational project. I think because of the situation we are in, there are more people talking about the provision of computational project, but how important it is. And do you, there in Edinburgh, do you run any lecture, training sessions in parallel to the projects?
No, not really. So in terms of university support for these types of projects, at Edinburgh it is pretty thin. So when I advertise one of these honours or master's projects, usually it says in the description, no prior programming knowledge required, but the student must have an enthusiasm or keenness to learn it. So often at the very start of the project, it's up to me to get them up to speed on the skills they need to start doing the real work.
Would it be possible to run this type of projects without much of the Linux, the Unix knowledge? Can they do more web-based?
Yes, I think so. So that was the reason really why I included this database of tools in my last slide. There are enough in there links to usually just web servers that pretty much anyone can use. You don't need to know Unix. You don't have to have a Linux machine. A lot of these tools are browser based, and if you can sort of collect enough of relevant tools, you could daisy chain them into a honours project, I think.
And then, of course, every time we're talking about a computational project and working from home, it comes to the mind. So there's some questions related to the bandwidth, the internet, how good it has to be for the student to be able to run a project like this from home.
Yes, so that is quite dependent on really the type of computational approach that they're using. So for molecular dynamics, for example, you can have extremely large data files. And for that, unless they do it all on a remote server, all the analysis, which is possible, if they want to download such a file onto their own computer, they really need a fast connection. But that's specific to molecular dynamics. For other projects like virtual screening, the files are much smaller, so you don't need fast fiber connection. And then for purely web server-based projects, you really wouldn't need a great connection at all. So, I mean, you would hope that they've got a connection that supports Zoom, at least Zoom with the cameras off. If they didn't, then I guess you could just use the telephone like we used to do. But yeah, so it depends on what you're hoping to achieve.
Right. So in terms of resources, Doug, there's some questions that people are asking, for example, for the flow chart on the resources. But am I right to say in this web server you mentioned that the link you provided, people can get enough information about, for example, which programs, softwares are needed and where to find them?
Yeah, I think so. So they're broken down by case use. So there are collected under headings such as bioinformatics, cheminformatics, binding site prediction, and things like that. The drawback to that website is that the list of tools is so long that you can spend a very long time looking through it, trying to find things that look relevant. So my suggestion would be to give that task to the students. That would be one of the very earliest jobs they could do after they've finished with all the reading, of course. They could come up with a short list of tools that they think might be useful, and then maybe you'd only have a short list to look through and figure out how to use.
Fantastic. So the other theme, Doug, is in terms of we discussed the student prior knowledge to do a project, a computational biology project. What about supervisors' prior knowledge? What supervisors needed to know before they propose a computational biology programming project to students?
So that, well, as I said, at Edinburgh, the official support for these sorts of things is pretty thin, and so for that, it did often come down to me to either know how to do these things, or at least know how to quickly find out. So that is perhaps a sticking point. So I mean, as an ex-protein crystallographer, I do know a lot about protein structure. So this simulating these peptides, it was pretty essential to have a basic understanding of why peptides do form alpha helices under some circumstances. I do know programming languages, and I do know Linux command line, and I know how to administer Linux networks and things like that. So I think in terms of that, again, you would want to stick to web server-based computational tools. Again, we go back to that. So for web servers, you don't have to be an expert in how the algorithm that's under the bonnet actually works. You just really need to have an understanding of what the input needs to be and how to interpret the output. But the better web servers, the output, they provide little reports for you, and they kind of give you final conclusions like, you know, these two proteins are significantly related or not. So yeah, I think there is you know, a certain amount of bottleneck there.
So then comes some questions which I think either of you or both of you, better saying, could provide some views on here. For example, at least lots of us, we believe that undergrad students really enjoy the lab projects, to be in the lab. Of course, the students who would take a computational project they probably looking for a different career. They wanted to learn something different. But do you, for example, run one-to-one sessions, weekly lab meetings with PhDs? Do you invite undergrad students to join to have that experience of being part of a group?
Yeah, I definitely think that's a good thing to do. If you have a group, whether wet lab or not, that meets regularly and perhaps somebody presents, then I see no reason why you wouldn't invite your honours project student to come along to that. And I think for a remote project, it becomes even more valuable, those sorts of things. So they feel more connected.
So now I think it is another interesting point in terms of the world we will be facing this online project. So how do we monitor individual progress remotely? So what do you do, Doug? And Vincent, what does he do that you really value? So not being able to see your colleagues and your supervisor and a face-to-face interaction as before.
I can say something about that.
Go on, Vincent.
Yeah. Well, I think there may be a bit of a, you know, Jedi skill that Doug has, because, you know, I think it takes some, you know, being a good observer from his side of what's the situation of the research, not to put pressure when it's not needed, and at the same time, you know, realize if I am not, you know, outspoken about it when we are getting stuck about things. And, you know, that's not maybe as straightforward as being in a wet lab, but there's still, you know, communicating efficiently, checking how things are going, and also like trusting that, you know, we are both on the same page and we are both enthusiastic about the project. So, you know, you don't want to be, I think, micromanaging the people, but at the same time, you want to make sure things are advancing. At least that's the feeling I get from Doug's mentoring.
Well, thanks, Vincent. I don't know about Jedi skill. That's just, I think that's just the beard.
So I think another important aspect is because of the situation we are and the way we are working now, we believe that students are having the opportunity to develop skills that perhaps we were not facilitating the development of some skills in the previous way when we used to work in the labs and everything face to face. If you were to identify some skills development, which ones would be? What do you think? Perhaps you, Vincent, because you were working in the labs, there came this change in the way we are working. So what skills you think were asked from you there?
Well, I think, you know, I think to me, everything starts with like being passionate about what you're doing. And then, you know, then it comes all the rest. Like, you know, if you have to learn and acquire new skills, and, you know, if that is programming, that is excellent. I think programming has become, you know, it's no longer something, you know, a specialty skill. I think it has become something that's a skill for life. And if you acquire it through these projects, like to me, seeing one of these programs running and getting reasonable results out of it, that is something extremely satisfying. And I wasn't thinking that was going to be the case when I started. Like I didn't know how to use the Unix command line and so on. And now I see that's something I could apply to this project, but I also could apply it to whatever I'm doing in the future. So just I think embracing the change and trying to find value in what you're doing and getting newer skills, I think that's something wonderful.
Yeah, I agree. I think it is really so. There is another question here, which it is, I think it's a bit generic. I'm not totally sure how much this goes with the computational type of project, but non-lab project, do you think would make students less competitive when applying for a PhD? Or this is really depends, as I said, the type of PhD career the students would like to have?
Yeah, I think it would really depend on the type of PhD they're going for. I mean, if they're applying for a PhD that is all wet lab work, and the student has not ever done a wet lab project, I am sorry to say I think that maybe would put them at a disadvantage. When you're hiring a student to work in a lab, you want to know that they can pipette accurately, for example. I mean, it's really important. So yes, but I mean, at the moment, we are doing what we can with what we have due to the current circumstances.
Okay, I just aware of the time. It's 2 minutes to 4. I didn't realize it went so fast. So I apologize if it's few questions that we didn't have time to address, but I think we covered most of the questions. So I would like to finish. Thank you, Doug and Vincent, again for a very useful webinar. And I hope that helped the participants on the planning of the projects for next year. So just before we go, I would like also to mention that we welcome suggestions for future topics and speakers for our webinars in the Biochemical Society. We will have a part 2 of this topic, which will be on the 11th of September. For more details, please do check our website. But before the 11th of September, on the 3rd of September, we will have another webinar, not related to this topic, but the title is Reprogramming of the Epigenome and Embryogenesis. Again, to register for our webinars, please do check our Biochemical Society. And while we are discussing remote teaching, there is also a website with great resources that have been created in the topic. It's lectureremote.com for those who are interested to get more tips about online teaching for next year. So thank you very much for all of you who joined us here today. And again, a special thanks goes to Doug and to Vincent. Thank you so much.
Thanks, Lou and Ellie.
Thank you so much. It's a pleasure.